Ladies and gentlemen, if we could all get back to our seats, we're going to start our final session of the afternoon. It's been a great day so far, and you'll, I think, be really delighted with our next speaker. So Dr. Ronald Cohn joined Sick Kids in 2012. Initially as chief of the Division of Clinical and Metabolic Genetics, he was recruited from the United States, where he'd already established an illustrious career. He started the world's first multidisciplinary center for hypotonia, earning both national and international recognition at Johns Hopkins. After arriving in Toronto and at Sick Kids, where he also is a senior scientist at the Research Institute, he became the inaugural Women's Auxiliary Chair in Clinical and Metabolic Genetics, but most recently, he was appointed as Chief of Pediatrics as the Hospital of Sick Kids, which would be the equivalent of the Physician-in-Chief position at UHN. Ronnie has a real breadth of interests, but what I really wanted him to talk about, which he kindly agreed to do, is the new horizons of gene therapy, which and the ability to do gene editing, which of course we're all familiar with. Ronnie, welcome, and thank you so much for coming.
Thank you so much for having me and giving me the opportunity to talk to you about what's not really my new passion anymore. It's the passion from the last 3 years. And I'm going to take you a little bit through a journey of how I got there and share with you some exciting aspects of our research and hopefully also engage you in a somewhat interesting discussion. One of the things I'm going to try to tell you here is that in my mind, although I'm a pediatrician and geneticist, I do think that genetic and genomic medicine is probably the most or the best example for a paradigm-shifting specialty that's impacting all areas of medicine wherever you look. And I'm not going to give you a full history lesson, but let's go through, since I know there are a lot of trainees here, let's go through this at least a little bit from a historical perspective. Everybody here has heard about the Gemmules theory of Darwin? Raise your hand. That's not as many people as I thought. It's just bad thing. So you should, I'm not suggesting that this is what you should read, but it's because it's hard to read. But Charles Darwin obviously brought forward the theory of evolution. And only a few years later, Mendel published his research on plants and peas, and we are still today using in clinic the term of Mendelian disorders when we are looking at autosomal or X-chromosomal inheritance of disorders. So even going back several hundred years, we are still using this term. Moving forward, Watson and Crick got the Nobel Prize for the DNA identification. Who was snubbed the Nobel Prize? Any other trainees? Does anybody know? So you should look that up. So Watson is known to be a brilliant mind and also quite a controversial figure. Has anybody ever heard Watson speak? Yes, so you know what I'm talking about. He is one of the most flamboyant anti-women and anti-race scientists who never makes a big deal out of his beliefs. And so despite his brilliance, he has now finally stepped back and is not talking to the public anymore, at least not in an official role as a scientist. Several years later, we had the publication of the human genome. One was supported by the National Institute of Health, and the other one was supported by Greg Venter, another brilliant scientist who is a very interesting character, who also published a sequence copy of the human genome. And something people may not know so much about 5 years later, we had the publication of the HapMap project, which was the first time that 274 individuals were sequenced in its entirety and really gave us an insight into where we are similar and where we are different in terms of the nucleotide changes and what makes you look different than me and you look different than you. And then several years later, we started to analyze these data, And what was a breakthrough for me and many others in the field was to recognize that all of us in this room carry a number of duplications or deletions, not just of a single nucleotide, but a whole chunk of a chromosome that doesn't appear to have any major impact on health. So we all carry some of these genomic rearrangements, and I think it's quite fascinating to think that all of us may be missing hundreds of genes or have a duplication of hundreds of genes, and we are still normal, whatever normal really is. But let me give you a patient example of the paradigm shift within one family. So this is Holly. Holly is a little girl who I met first when I was at Hopkins. And I was a young attending, and I saw this child on a consult service when I was actually the pediatric ward attending, because she presented at the age of 3 months with severe seizures. And I threw every kind of genetic test at her that I knew of. It took me over a year to get all of these done. It cost about $15,000. And I couldn't find an answer for her epilepsy. And she really started to become more and more progressive. And shortly before I moved here to Toronto 5 years ago, she passed away. And I remember I went to the funeral, and the mom said that we were very close in trying to keep her as comfortable as possible towards the end. But the one thing the mom said at the end was, if I only would know why Holly was sick. So several months later, she called me and said, I'm pregnant. And I was very excited. And again, several months later, she called me that her daughter was born, but she started to have seizures. So this is Alexa. It was very obvious that at this point that she had the same disease like her sister. Now at this point, we were able to now sequence the whole genome, and I offered mom the opportunity to do this in order to try to find out what's causing the disease of her child. And we did make a diagnosis in a gene called PIGN, and don't feel bad if you never heard about this gene. I never heard about this gene until we found it. And interestingly enough, there were only 3 families reported in Israel who had a mutation in this gene. And now we know about 15 families all around the world who have this. But I want to tell you that probably one of the most humbling aspects of my professional career so far has been that I honestly underestimated the value of providing families and parents an answer to their question of a diagnosis. Because in this case, we found an answer. I don't have a treatment. I can't do anything. The family is very religious, so I can't even offer prenatal screening. The mom actually got pregnant again and called me, and I said, we can't do prenatal testing. And she laughed at me, knowing you know me, that I wouldn't do this. And I knew this. She had a healthy boy afterwards. But the amount of relief this family, and over the last 5 years, many other families have found in us just providing them an answer to why their child is sick, is something that I really honestly have underestimated and has been probably one of the most humbling experiences in my career. So what if we could begin to think about treatment options for all genetic disorders and trying to develop a concept of how to do this? So I would like to introduce you to the field, if you want to say so, of interventional genomics. I didn't patent this. We came up with this in my laboratory. What it really is I think it's starting to finally develop a concept for how to repair a genetic mutation. And I'm going to show you a little bit of data of how to do this. And all of this is going to come through CRISPR. Is anybody in the room who has not heard about CRISPR? No. So you see, that's amazing, because 2 years ago, when I gave a talk to students who came to Sick Kids over the summer, nobody had heard about it, maybe a handful of people. The following year, everybody knew about it. So the way how I learned about CRISPR is that I moved to Toronto, and the first paper was published in Nature by Jennifer Doudna's group, together with Emmanuel Charpentier. And I think you agree with me that sometimes it happens to you too, that you find a paper, you think it's interesting, you put it on your desk, and then you think you need to read it, and you don't. And then several months later, I had a science paper coming out from a different group, Feng Zhang in Boston. These are the two who work together but are also competitors. And I put it again there. Then I went on vacation and took the papers with me. I'm not such a geek that I always do science on vacation, but I thought it might be a good opportunity. And I didn't understand a word. New language, it was not the right thing. And then this paper came out several months later. It was not a paper, really. It was just a report in a late-term newspaper like the Time magazine of England about this jaw-dropping breakthrough hailed as landmark in fight against hereditary diseases. And within 24 hours, I had close to 40 emails in my inbox from families of patients of mine with all different kind of disorders asking me, is this going to be one day something for my child? So that was kind of the necessary push I got to finally sit down and read this. And I've been honestly mesmerized ever since. I went to my research associate, showed him this work and said, do you think we can do this? Do you think we can look at this from a therapeutic point of view? He was as excited about it as I was, probably more so, and he's much smarter than I am for sure. And that's how we changed the research in my laboratory. So the young students here who always hear from your professors, stay focused, focused, focused. That's true. Sometimes following your heart and passion is a good thing too. So that's what we did. We changed research in my lab. And over the last couple of years, you can see that CRISPR has been in the news everywhere about different things, also in Germany and many other countries. And I want to give you a little bit of just a high-level idea of what's the difference between CRISPR genome editing and other therapeutic approaches of altering the genome. So let's take the sentence, twinkle, twinkle, little star. And this sentence is now mutated with a mutation making it twinkle, twinkle, big star. So when you do gene therapy, what you do is you are offering the body or the cell an additional gene to correct this mutation, and it looks like this then, twinkle, twinkle, little big star. So you're giving back the little, but you're not really fixing the big, and that's what gene therapy is. There's the idea of RNA inhibition, so you can inhibit and remove the mutation, which makes it twinkle, twinkle. But if you actually do CRISPR genome editing, you are able to restore the full sentence to twinkle, twinkle, little star. So that's the general idea and the advantage of CRISPR. The beauty about the system, or what I find fascinating, is that this wasn't invented by anybody, but it was invented by evolution. So it's a bacterial defense mechanism that has been around for millions of years, where bacteria protect themselves from an infection of a virus. So these viruses want to attack the bacteria, and the bacteria identifies a part in the genome of the virus to cut out a piece of DNA and thereby inactivate the virus. And why do we need bacteria to do that? Anybody? The room is too big for interaction, I guess. Yogurt. You're the first in many, many lectures all last year who says yogurt. Finally, somebody eats yogurt and healthy in the morning. So there are many, the microbiome is another example. So that's why evolution has invented this. And what Jennifer Doudna and Emmanuel Charpentier and Feng Zhang really figured out is for what this bacteria can do to the virus, we can do anywhere in the genome. And here you have a video that shows you how this potentially looks like. So this is a microscopic high resolution of a cell where you see the DNA, and you see these purple creatures, which are the guide RNA. So that's the first new word you have to learn in the language. And they find a specific region in the genomic DNA, bind to this, and then you have the Cas9, which is actually an enzyme that comes in and makes a cut wherever you design the guide RNA to bind. And then you can insert a piece of DNA, although I will tell you later that the insertion of the DNA is quite a sophisticated thing to do and is quite difficult to accomplish. So what can CRISPR do? CRISPR has already revolutionized the way how we do science. everybody, like I said, you all know what CRISPR is because you're probably using it. I mean, we are probably using it already the way how we use PCR machines. You can create an enormous amount of animal models now in several months. Has anybody in this audience ever tried to breed a triple knockout mouse? Ha! How long did it take you, Mansour? 9 months to breed? Okay, so he's just inflating my story. When I was in Iowa City and I tried to breed it took me 2.5 years to finally get the triple knockout mouse. Now you can create a triple knockout mouse within 5 months. Still faster than Mansour can do it, but not as much faster than I could do it. The other thing which CRISPR can do, and I will show you some data about this later, is you can inactivate the Cas9 enzyme. So you're not actually cutting anything, but you can direct the guide RNA and change expression of a gene by either upregulating it or downregulating it, which I think has maybe even more therapeutic benefits once we get to fine-tune the system than the genetic repair. But what I'm going to focus on is trying to identify mechanisms where you just have to cut out a piece of DNA and thereby fix a certain genetic problem. Because if you want to cut out a piece of DNA and then insert, like on the video you saw, a synthetic DNA with a corrected sequence, you are dependent on a specific time in the cell cycle for that to happen. So any kind of post-mitotic tissue in the body, for sure, is not going to work. So as you know, or as Mansour told you, I'm mainly interested in the nervous system and skeletal muscle. So these are both post-mitotic tissues. If you deal with liver or lung or skin, it's different. So you might be more efficient. But we have really focused our efforts to try to find ways to just cut a piece of DNA out and make things better. The first example I want to show you is achondroplasia. It's probably a disease you all heard about. It's called by one mutation. All the patients, or 98% of the patients have the one mutation in the FGFR gene, FGFR3 gene, and it's a dominant negative mutation. So the idea would be, can you remove in an allele-specific way that mutation and correct the disease? And that's exactly what we were able to show here. You can see this on the graph below. We designed the guides in a way that it would just target and cut the allele with the mutation. That's not going to work for every dominant negative allele necessarily, although the system has been developing and improving quite a bit. But you can design it for about 80% of single mutations in a way that it only cuts on the strand where you have a mutation, because you need to think about that the rest of the pieces of the DNA are the same on both strands. So you need to make sure that you don't cut on both sides. So we have done this, and there's a postdoc at SickKids who is interested in this, who is going to take this further into an animal model. But the next group of disorders I really want to focus on are duplicated copy number variations within the genome. So if you think about the idea, if you have a duplication, then you see here the AB is the wild-type gene, and another AB would be the duplication of the gene. And the initial idea would be you cut out just at the junction with one scissor, one guide RNA, and another scissor a little bit outside of the duplication, which would remove the duplication and give you the wild type, right? But what's not so good about this? Two things. A, if you think about how to package two guides into a delivery vehicle for systemic delivery, two are always more difficult than one. And if you think about off-targets, each scissor comes with its own risk of off-target mutations. So the brilliant research associate I talked to you about earlier in my lab figured out you can just take one scissor that cuts in the intron of the wild type and the duplicated area, which is the same because it's duplicated, right? And then you create the wild type situation like this. This is a little sentence of a video that shows you how to do this. So add one diced avocado and 1 teaspoon of salt and 1 teaspoon of salt, and you're trying to hit the end which is both in the wild type and the duplication. You remove it and you have the full sentence. So this is a patient of mine, Eddie, who I saw, one of my first patients actually when I moved here to Toronto, who has a duplication of the MECP2 gene, which is going along with low muscle tone and developmental delay. And the family approached me about this because they heard about this in the news, and we just changed research in my lab, and I said, you know what, we can try to do this in the fibroblasts of your son. So we designed guides. And as you can see here on the lower side, you see two bands of the duplication. Let me just show this to you here. So this is the wild type, and here you see the duplication. And when you give the guide, you remove the DNA on the duplication on the DNA level. And we are in the process of generating an animal model with exactly this duplication. And I'll talk to you about the complication of this in a minute. To see whether we can do this in vivo in a patient model for this disease. So let me talk to you about a different disease, which is a disease I've been working on for 20 years, which is Duchenne muscular dystrophy, which is one of the most common forms of muscular dystrophy. It's associated with the loss of dystrophin. So you can see here that this is normal muscle architecture, and all these green circles are dystrophin-positive fibers. And here you have significant fibrosis and no dystrophin, and that's really the hallmark of this disease. And I want to introduce to you this young man. His name is Gabriel. I met him when he was 4 years old. And he's really actually not a patient of mine. He's the son of one of my closest friends in the UK, in London. And I met him through friends, and they wanted to give me money for my research 10 years ago. And I said, no, I don't want to take your money. And I became the chief scientific advisor for the foundation. You know, sometimes you click to people, and we became very close friends. He has an interesting duplication in the dystrophin gene. And we always try to support research all over the world over the last 10 years that would somehow be applicable to him. But there was never really anything that was quite as close as what I'm about to tell you. That's Gabriel today, 10 years later, 11 years later. And as you can see, he lost his ambulation. He's sitting now in a wheelchair. And although he's a happy camper about it, the disease has caught up to him quite significantly. And one day I called his mom. Like I said, she's a good friend of mine. And I said, you know, after we finished the experiment with the cells of the MECP2 patient, I knew that we could do it. And I called the mom and I said, we want us to do this in Gabriel's cells. And so we had an hour conversation about what if and what there. And at some point I said, I can't answer your questions anymore. She's incredibly smart. And then she stopped and she said, you know, Ronnie, if it works and you're going to make a movie out of it, I want to be played by Angelina Jolie. So that's the way how this mom is. And about a year ago, somebody suggested Liam Neeson for myself. I'm not that old. So anyway, so we got his cells from London. We did the experiments. Here you see the duplicated band. It's a different assay, so the duplicated band is a lower band. And as you can see, we gave the guide to the cells, and we were able to remove a significant portion of the duplication. But what is it you really want to see? You want to make sure that this is going to make full-length dystrophin. So we took the cells, and here you see wild-type dystrophin expressed, and under GFP you see Gabriel's cells without the guide, and if you give the guide, you get dystrophin expression back. And you get dystrophin expression back at the full-length wild type. So I don't know how many people in the audience here are following the Duchenne field, probably not many, if any. But all the current therapies, including gene therapies that I'm very excited about, they all give back only a short version of the dystrophin. So this is actually the first time that somebody was able to find a way to replace the whole entire wild-type dystrophin. How much you need of this? I don't know. It's a good question we can talk about during the discussion time. So we took now other cells of other patients with duplications, specifically these 3 to 20, 2 to 6, 6 to 7, because all of those would be able and amenable to the same guide. And we're going to talk about the complexities of individualized treatment as well. And we were able to remove the duplication in all of those with one guide, which is important because here you have one guide that can treat several patients, because Gabriel is the only patient with a known duplication like this. I've never met anybody else with that duplication. So what's the next step? Anybody or the students wants to offer? What should we do next? So it took us 8 months to figure this out, so I'm not expecting an answer in 20 seconds. But we figured out that CRISPR, whenever you start to cut within the genome, doesn't necessarily just cause deletions, but can also cause inversions and duplications. At a lower rate, but it has been shown by other groups while we were thinking about this that you can probably use the CRISPR system to create a duplication mouse. And that's what we did. We generated Gabriel's mutation duplication in a mouse model. You can see this here. And the mouse does have muscular dystrophy, as you can see here. It does not express any dystrophin. It's significantly weaker than you would expect. And we were all excited because we had the model. And now I talked to Health Canada, and they said, get a mouse model, and if you treat the mouse model, then we can sit down and talk about how to move this forward. So we injected the guide into the mice, and nothing happened. We did it again and again and again, and we couldn't remove the duplication. So it turns out that, so this is for the students here. So far I've shown you everything going one by one beautifully. And I have to say, 3 years ago we started this. I'm going to show you more data. We have had a lot of luck, but this one didn't go the way how we wanted. So I'm not going to walk you through 6 months of unbelievably tedious work. But what I'm going to show you is that these mice have a duplication and lots of other rearrangements in the dystrophin gene too. So we are actually able to remove the duplication, but the reason why we don't see dystrophin is because there's so many other messed up things like an inversion and other duplications and deletions, which is shown here. And again, I'm not going to walk you through this too much because my postdoc, my student who did this work has lost a lot of tears and sweat over this work. And what is difficult about this, what made us nervous is this something that we created with CRISPR, or is it actually something that happens in all patients who have a duplication? Because nobody ever sequences the entire gene the way how we did with whole genome sequencing. And although we were able to remove the duplication in Gabriel's cells, we had to go back to the patients and sequence the gene to make sure that these are clean duplications. And I can tell you the good news about this is that, yes, they are all clean duplications. So the idea we have to remove those is still valid. We just have to get an animal model to prove that it works in vivo. So here are 2 strategies we are currently undertaking, how to rescue the current duplication, because one of the mice has only, quote, an inversion in addition to the duplication. So what we're doing on the left side, you can see, is we are hoping that through natural recombination, we can get rid of the inversion. That's about a 2.2% chance of happening. That's the way how we calculated it based on the distance. So we have bred about 500 mice. So far, it hasn't happened yet. And now the other thing we are trying to do is we're actually trying to remove and flip the inversion with CRISPR to leave the duplication in place. And here I can show you some promising preliminary data that this probably worked in this mouse. Here we removed the inversion and we have evidence that the duplication junction is there. So we're breeding this mouse now, hoping that we can create the animal model around this. But I think what's important here is you have heard me talk about my patients and my friends. And every 3 or 4 months, I do ethical rounds in my lab when we have new students coming. Because this is the most individual type of science I've ever done, and I think one can probably do all together, right? And I would say, no, I can say that 90% of the people in my lab, 9 out of 10, get an extra boost of doing research on patients that I know or that they even have seen in clinic with me. But some feel the pressure of this, and this student who is working on Gabriel, who knows that time is running out for him and that we don't have 5 to 10 years to figure this out because he's going to lose more and more muscle mass. While she never admits to the fact that this is high-stake pressure, I know it is. It has to be. It's for me too. And I think we just have to recognize this and have these open discussions around this because it's the way how science is going, and if you are going to enter this field, and I hope many of you will, not just because of this talk, but because you think that there's validity in many of these disorders, it's an additional layer of complexity that's positive, but can be negative at times. So I'm going to show you one more application, a different form of muscular dystrophy. This one is a little bit more rare. Here you see a patient of mine from Baltimore still. Him and I Skype about twice a year, and we are vivid Ravens fan. Anybody a football fan here? No? No, Canadians are not really football fans. That's fine. And him and I talk about football. The only bad thing about him is that he's a Trump supporter, and so we don't talk about politics. So it's caused by a mutation in the LAMA2 gene, and there's a natural animal model, as you can see in this video. And I want you to pay attention to this mouse on the right side, on the left side, that's really paralyzed in its hind limbs. That will become important as we move on. So we use this natural occurring mouse model for initially the hypothesis to modulate the expression of a gene that has been shown to be a modifier of this disease called laminin alpha 1. So I told you earlier you can use the CRISPR system to also change gene expression, and here I'm going to show you some data about how we did this. So laminin alpha 1 is a homologue of laminin alpha 2, and there were plenty of studies of transgenic mice that have shown that you can correct the phenotype by overexpressing this gene. So I had a postdoc in my lab who did this, and you couple the guide RNA to a transcriptional activator domain called VP64. And with this, you can then modulate the promoter of the gene and lead to increased expression of the gene. So we did initially an intramuscular injection just to make sure that the system actually works. So we injected the mice into the tibialis anterior muscle. And as you can see here, the negative control doesn't express laminin alpha 1. And if you use one guide, you get quite significant expression in the injected muscle. And if you use 3 guides, you also get expression, although it seems that the expression is not much higher with 3 versus 1 guide. So we, in the following experiments, we went to use only a single guide. If you look at the expression of the protein, you see that all the protein is overexpressed the way how you would expect it. And what you though want to do is, for muscle diseases, you can't think about intramuscular injections for young patients because you can't do thousands of injections to the muscles, right? So you need to look at systemic injections, and the vehicle you can use for skeletal muscle as well as heart muscle and the nervous system is an adeno-associated virus type 9 that has been shown to transduce all these tissues quite well. So we took the guide RNA into the AV9, and we did systemic injections to the tail vein of these mice. And you can see that at a lower dose, you get a little bit of expression of laminin alpha 1, but at a higher dose, which is still within a so-called therapeutic dose, if you would extrapolate this to patients, you get quite robust expression. And I'm not sure how well you can see this, but you get quite significant improvement of the muscle architecture. So you can see that this area here looks a little bit less homogenic than this. And this is an area that doesn't have as much expression. So you don't get 100% expression, you never do. But it correlates really nicely. Wherever you get expression of laminin alpha 1, you correct the phenotype, and wherever you don't, then the phenotype is still existing. We quantified other things like centralized nuclei and fibrotic areas, and you can see that there's significant improvement. There's improved contractile force if you do like an EMG type of assay in a mouse. And here you can see a video. This is a non-treated mouse who is still limping. And that's a treated mouse. And what you can see in a minute, this mouse is able to be vertical, right here. So that's something the non-treated mouse is never able to do. So we took this video only 3 weeks after treatment, and we are now collecting data up to several months to see whether the effect is sustainable and actually improved. It looks like that the effect is actually improving over time. So I'll have more about this next time maybe. So just to conclude on this part of the talk, we had the catalytically inactive Cas9 coupled to the VP69. It was a successful modulation of laminin alpha 1. And the important aspect of this is that it's a mutation-independent approach. So each patient who has this disease is potentially amenable to that therapeutic approach. So the last few minutes, I want to show you the work of a postdoc of mine, another brilliant work, which again is trying to take advantage of the fact, or trying to take advantage of the disadvantage of a postmitotic tissue where we cannot fix a mutation by simply inserting a DNA. So these mice, which I showed you earlier, have a splice site mutation, and so do about 40% of patients who have this congenital muscular dystrophy. And the splice site mutation is a mutation that loses exon 2, and my postdoc was thinking maybe we could use CRISPR to find a way to include exon 2 back into the gene and lead to wild-type expression of the gene and protein. So you have this G to A splice site mutation that leads to a truncated form of laminin alpha 2 at the end terminus. And the way how she thought about this is the following. If there is potentially a consensus motif for an alternative 5-splice mutation site, then you should be able to cut a little bit outside of this intron and a little bit further downstream, take out a piece of the intron and create and pretty much trick the cell into a situation of restoring the exon 2 by thinking, oops, the splice site machinery should get active. And that's exactly what she did. So we designed 2 guides. We injected this in newborn mice into the temporal vein right here. And this is how it looked like. So under GFP, you don't see any inclusion. You can see here that the injected mice have evidence of exon 2 inclusion. We sequenced this exon. It was fully intact, fully the way how it was supposed to be. When we looked for protein expression on the Western blot, you can see that the guides lead to restoration of protein expression. You can see this here on the GFP versus guide immunohistochemistry. And on the left side, you see the fibrotic staining, which is significantly improved. So you have less fibrosis in the muscle after treating these mice. We also were able to hit the peripheral nerve, which is also affected in this disease. And you can see that after CRISPR treatment, we have a restoration of myelination in the peripheral nerve. And we did a number of in vivo assays to demonstrate functional improvement. So here we were looking at how much the mice actually walk in terms of total distance, and you can see that the treated mice walk almost as much as wild-type mice. If you look at this vertical activity, which I showed you earlier, it's almost as much as wild-type mice. And if you look at contracture of the muscle, it's, again, very close to wild-type muscle. And here's the video to to demonstrate this. So here you have an older mouse. That's a 9-month-old mouse. And that's the treated mouse. So I actually asked my student to repeat this 3 times before I wanted to believe it, because it was such a striking difference. But as you can see, we were pretty excited about this. We were proving our hypothesis, and what we are doing right now is we have collected 5 patient samples from all over the world of patients who had either muscle biopsy or a fibroblast biopsy, who have a proven splice site mutation, to see whether we can do this in vitro in these patient cells, and then begin slowly conversations about what are the next steps, and we can have this conversation together in a few minutes. I want to end with a little comment about that. I think we have to challenge the culture, how we think about clinical trials and drug approval for genetic disorders. So what I've shown you today is many of these therapies will be individual therapies. There might be an N of 1 only. And how are we going to regulate something like this? So I think we have to start getting Health Canada, FDA, and EMA all involved in discussions around we can't run clinical trials for any of these disorders. And to be honest, given the complexity of what we know of generating animal models for complex rearrangements, we shouldn't be expecting to make an animal model for each duplication or each deletion we are showing, right? And I can tell you, Health Canada has been incredibly open to these conversations. They have been willing to listen and said, as soon as you have enough of these data, we'll sit down with you and think about, similar to how we do research in oncology, where there is a lot of N of 1 type of research, we will be willing to think this through with you. On the other side, you have to think about how is industry going to play a part in this? Because who is going to be interested in developing a therapy for one patient that, in theory, might have to be applied only once if it works? How are you going to price this? So just out of curiosity, tell me what you think. How much would it cost to actually make this drug for a muscular dystrophy patient like Gabriel? How much do you think it would cost to just you put this in an AV9 and you give enough at a dose which is high enough to potentially get some results? How much do you think it costs? Give me a number, anybody. Okay, millions of the R&D, but to actually just make this, it's $150,000, nothing, right? And to be honest, if you think about how much work has gone just into this particular project, it's not millions of dollars. We didn't spend millions of dollars to get to the point where we are right now. Maybe we are approaching $1 million soon. So I think we need to have conversation with industry too, because we have all these exciting treatments coming, and we need to find a way to have them help us develop them and also price them in a way that it's not going to corrupt and break our healthcare system. Has anybody heard about Spinraza, the treatment for spinal muscular atrophy? Go look this up on the Google. That is a home run treatment for kids who were never able to sit and walk and move. They can now sit and walk and move, but it's priced at about $1 million per year per child. Can't do it. We can't do it like this. So I'm bringing this up because I think a lot of interesting discussions have to happen over time. This is the wonderful group of people in my lab. Let me just quickly point out. So this is Zhenya, who's kind of the brain behind everything in my lab. This is Dwi, who did the work on the laminin alpha 2 mice. This is the student who is sweating a lot about the duplication mice, and he did the upregulation in the laminin alpha mice. And I leave you with this quote from a postdoc who said, "I'm not sure what a golden age looks like, but I think we are in one," and I would agree with him. Thank you.
Thank you for that talk. It was very interesting. So my research and clinical focus is genetic kidney disease, so I follow this related literature. A couple of years ago, Eric Lander wrote a letter in New England saying that he thought CRISPR technology would have limited therapeutic applications because you could just do pre-implantation genetic diagnosis to prevent transmission of a Mendelian disease. So I'm just wondering what you think about that.
Thank you for bringing this up. This is terrific. So what Eric Lander wrote about is the entire issue and complexity around germline editing using CRISPR. So everything I talked to you about today is what we call somatic cell therapies, meaning you're treating a patient and the patient only. Eric is talking, and he's one of many thought leaders who is talking about why should we take the risk of editing the human embryo before we know what potential side effects can happen. So if I do something in a living patient and something goes wrong, it's terrible and I don't want to even think about it. But if I do something in an embryo and it has an unintended consequences, then it's for generations to come. And before we really know that the system is safe, we shouldn't be thinking about eliminating genetic disease through germline editing. And his argument is very well taken. It's the same argument I've been using, Janet Rosant is using when people ask us, we're doing PGD, there is no real difference. Why would you take the risk? People who are for germline editing say, yeah, you don't have to take less hormones and you have more eggs available. This might all be true, but that compared to the risk of affecting generations to come with an unintended consequence, it's just not there.
Ronnie, you mentioned the challenges of somatic cell gene editing. Can you say a few words about the number of cells that your AAV approach for muscular and neural tissue targets? What's the transduction efficiency? And then also, can you just share with us your thoughts about non-somatic cell, epithelial cells, whatnot, that may have mutations that are functional? How the targeting of them, what are the other issues there?
So from experiments in mice and dogs, we know that we target about 50 to 80% of skeletal and cardiac muscle. How this translates into humans, we actually don't know. So I'm associated, I'm working with a company on a microdystrophin gene therapy trial that is going to use AAV9, and we're probably going to inject the first patient towards the end of the year. The patients will tell us whether we get the same transduction efficiency. And if we do, then it's another home run. I expect it won't be quite as much. You don't need as much. So what we do know is in skeletal muscle disorders, you probably just need to hit about 15 to 20% of the cells to have a therapeutic benefit. For the heart muscle, we don't know quite how much you do, but it's probably not much more than that. Epithelial cells are not post-mitotic cells, right? So here you can actually use the system and try to
target a progenitor.
Target a progenitor cell and insert directly the corrected version of the DNA in a much more efficient way. And there are AVs and other nanoparticles who can deliver it to epithelial cells much better than to muscle or nerves.
Any other questions for Ronnie? Ronnie, one other thought occurred to me when you mentioned AAV. You'll remember the early days of gene therapy trials were confounded a bit by the viral reaction to the viral vectors, the immune reaction to the viral vectors. What's the status of that with the AAVs? And does that limit efficacy?
So first of all, what Mansoor is talking about is that about 15 years ago, people used not adeno-associated virus, but adenovirus as a delivery vehicle to treat a metabolic disease called OTC deficiency. And the two boys died. And it paralyzed the gene therapy field literally for 10 years. Now with adeno-associated viruses, We believe that the risk is less, and I can tell you for AAV9, it has been used in clinical trials of 3 different disorders, and it appears to be safe. So the issue here, the major issue with this one is only twofold. One, there's a number of people out there who are pre-immunized against it, so have antibodies. And there's a lot of research going into how to mitigate that problem. And the second problem is, although in theory, 1 injection should be sufficient, we probably believe that over time you're probably going to lose this. And once you inject it once, you develop antibodies against all of the AAV serotypes. So we have to find a way to overcome this. Because if you ask me today, so when this mother of Gabriel asked me, so what do you think is best case scenario for him? I tell you what I dream about when I go to bed at night, that he's going to get this gene therapy treatment at the end of the year, and we're going to give him the CRISPR treatment in 3 or 4 years on top of it, but he can be stabilized. So we just have to find a way to overcome the immune reactions to that.
All right.
Thank you.
Well, if there are no other further questions, Ronnie, beautiful lecture.
Thank you.
Thank you so much for bringing us these insights. So if I could get my slide, I'm going to, one of my final tasks for today, and one of the most enjoyable, is to recognize some of our trainees. Do I have to move it forward? Or? So I think I'll just, without having to necessarily put up a slide. Oh, that's okay. That's all right. I'm going to invite up if they're here still, and hope they are, the people who've won our postdoctoral fellowship awards. Every year we have a competition. It's a really tough competition that the leadership group and myself have to sort of choose from amongst really high caliber of trainees. And we're really pleased again to give out 3 awards this year. So I'm just going to mention them all, and if they could come up and receive a certificate, and we can give them a round of applause. So it's Zahra Muhammad Ali, Peter Di Stefano, and Junichi Sugihara. Congratulations. Congratulations. Oh, they want to get a photo? Okay. Peter? Congratulations. Okay, we'll get a little photo. Thank you. All right, and then, as I mentioned this morning, we had from the long list of abstracts presented, a tough choice in selecting our 6 moderated finalists. So I'm going to invite all 6 of them to come up because they all get a prize. It's Laura Appel, Lindsay Wu, Yaseman Agadaze. And then we have the 3 who get slightly bigger prizes, Dakota Gustafson, Paige Bauer, and Doreen Zarinchat. Congratulations to all of you. So I have here, this is always fun. We don't need a drum roll or anybody, but these are tough, and I want to thank the judges for going through this. But I have in 3, 2, and 1 order Dakota. Congratulations. Paige and Doreen. Congratulations to all of you. Your photo. All right, well, that brings the day to a close. I want to thank everybody for attending. I hope you've enjoyed the day as much as I have. The theme was integrity and innovation, and I think the integrity, I couldn't ask for a better lecture than Eleftherios' lecture this morning on the reproducibility or irreproducibility of science and the role that we all have to play from every level, right up from trainee to PI, on thinking about that, reflecting on that, as he said. And then on innovation, again, my thanks to all the speakers about their innovative work, and particularly to Professor Cohn for bringing us an update from his own lab, a very personal update with pediatric patient populations and really a new horizon in medicine, which is genome editing. So thank you very much. We will be sending out a survey, which I hope you'll all respond to, about how you liked the venue, what you liked about the day, the forum. We do take your feedback very seriously in an attempt to do better every single time. So thanks very much, everyone, for attending.